just getting ready to go live. So yeah, okay, that's fine. Thanks, Sarah. Okay, Chris, whenever. Welcome, everyone, to Casual Night with Arachnids at Ohio State University, Newark. We are pleased to have an excellent speaker series here for the evening. We're going to talk a whole lot about different arachnids. You guys should learn a whole lot of things that you probably never knew about spiders and scorpions and creepy crawlies, oh my. We have a chance to take questions both from our live audience and also from the streaming audience online on Science on Google+. So we will try and take a few questions from each. I'm going to introduce the first speaker. Our first speaker for the evening is Dr. Doug Gaffin from the Department of Biology at the University of Oklahoma. He is a renowned expert in scorpions and is going to talk to us a little bit about how to read their minds. Doug?
All right. Thank you so much, Andy, and thanks everyone for coming out. I'm Doug. I'm from University of Oklahoma, and I want to share with you an animal that might be somewhat unfamiliar to you. It's called a scorpion, a type of an arachnid. And so we have a Spock theme going on here. And so as we approach scorpions, scorpions evoke lots of emotions, not the least of which, of course, is fear. And so it's very important to take a very dispassionate approach, maybe like Dr. Spock might take. So here we go. First officer's log says we find ourselves in the sand dunes of the Mojave Desert where we have discovered 10 eight-legged animals with fascinating habits. When we approached these creatures, they attacked us with their tails. But we obeyed the prime directive, resisted the impulse to step on them, and simply observed their activities. And the first thing we observed is this. This is a scorpion under daylight. And then we turn out the lights, and we turn on a black light, like I have in my hands right here. You will see this. So I have in my hands an emperor scorpion fluorescing under an ultraviolet light. So for the people at home, you can see that when I take it away, it does not fluoresce. When I bring it back, it does. So the entire scorpion's cuticle, its skin, fluoresces this bright cyan green under fluorescent light. The second thing, all throughout the southwest of this country, there are just wonderful ecosystems. I love sand dunes all the way from Texas to New Mexico to Arizona out to California. You'll see places like this. And if you get out of your car and look really closely, you'll see little slit-shaped burrows. There are scorpions down there underneath the sand. They go down about a meter. They are nocturnal, and they come out during the night. So you don't see them during the day. But if we're real quiet, maybe one will come out for us. Here it comes now. It's coming out of its burrow, and it walks around the sand, and it stops. And it walks a little further, and it stops, and it sits and waits. They are very patient. They're waiting for little crickets and insects to walk by. And they actually hunt by vibrations. They feel the seismic spread of the little cricket's footsteps. And that's all it takes for the scorpion to find the cricket, to capture the cricket, to sting the cricket, and to eat the cricket. And then before the sun comes up, that scorpion will make its way back to its burrow. And in these sand scorpions, the males and the females, they maintain their own home burrows. And so time goes by, and here's this guy in the bottom of his sand dune, and he's thinking, all right, there's January, there's February, March has gone off, April, May, June, July. Well, now it's August, it's time to mate. So the male will leave the comfort of his burrow and start walking across the sand. So here's a female, she's emerged from her burrow, and what we think is going on, we think that she's putting down a chemical, some sort of a chemical on the sand. It's called a pheromone. And so if we're real quiet, maybe we'll get lucky and find a male who's looking to find her. Here he comes now, right there. He's walking across the sand. And when he's near to where the female is, when he's over where she put this chemical down, he can't control himself. He does this little juddering dance. Here it is right here, a little jerky behavior. It's called juddering. It's a pre-courtship behavior, and if she's receptive And what I didn't tell you is that these animals are cannibals. So the females will eat a lot of the males. But if she is receptive, she will turn and allow him to approach her. And he'll grab her claws called pedipalps. And then they engage in a really nice dance. It's called the Promenade à Deux. It's very romantic. It's under the starlight. It's really not that romantic. He's actually dragging her along now. And if he encounters a little twig or a stick or a rock, he will survey that with some organs on his belly and release a sperm capsule, a little piece of skin of cuticle that has sperm in it, and it's got a sticky end on one side, and it's got a little trigger on the other. And then he carefully maneuvers her over that little trigger, and it shoots the sperm inside of her. Fertilization takes place inside of the animals. They hatch out inside, and then he's done. But a year later, then the babies will crawl out of the female, and this is what you might see. Isn't that cute? A bunch of little scorplings on the backs of a scorpion. This is a different species, but nonetheless, they really are kind of adorable. Well, I want to show you, and if Terry could help me here real fast, if you could just call up this YouTube video real fast. What you're going to see, we'll just click that, and if you can give me some hits on YouTube, that'd be awesome. But this is an old experiment I did a long time ago. I put a chemical wash from a female up near the top of the screen. This is a male who just woke up. He's going to walk over where the female chemical is, and he's going to get very excited indeed. He's now just reached the area. He backs up. He does these little push-ups. You may see these little organs. Those are called pectins. Remember that. Those are brushing the sand. He does a really strange behavior. We don't know what this is about, but he actually starts wagging his tail right there. That's interesting. Who knows what that's about? But he's a terribly excited animal. He thinks there's a female here, and he actually does these judders I showed you before. This gets more and more intense as he gets nearer the middle. He's very confident there's a female in the area, but he just can't find her. Terry, can I get back to the PowerPoint here? So that's called juddering. It's a pre-courtship behavior, but you have to have a way to pick up the chemical, right? And you saw those little brushes the animal was swinging across the sand. Those are called pectins. And next time you see a scorpion, just watch them. They're on all scorpions. They hang down from the bellies of all scorpions. When we take them off the male doesn't do that behavior. So we think this is a very important organ for picking up that signal. Well, let's go into these organs here. Let's see what they look like. There's a whole world of exploration. They're paired comb-like structures from the belly of all scorpions. Let's look closely at one. It's got a flexible spine and a series of teeth. Let's get very small. I flipped it over on you. This is under something called a scanning electron micrograph. And you see on the tips of each of the teeth hundreds of little tiny dots. Let's look at one tooth. Oh, those dots turn into a field, a forest of pegs. Those pegs are called peg sensilla. That's the business end. That's what's actually doing the tasting, if you will. Let's look right down on the top of a patch of those pegs. This is the way the ground would move as the animal brushes his pectins across the sand. Let's look right down on the throat of one. There's the pore. If you're going to be a chemoreceptor, you've got to have a way to get the chemical into your neurons, and that's it. Let's take a cross section. You'll see there's actually a whole bunch of chemosensitive cells in there. There's actually like 10 to 15, and there's one mechanosensitive cell that's responsive to the bending of that little tiny peg. And what we do in my lab is we take little tiny electrodes and we put these tiny, tiny electrodes through the bases of individual pegs while blowing chemicals across the top. And then we can get a sense as to what they actually are thinking. What are they tasting? What are they detecting? And this is what it looks like in my lab. There's a big old microscope. There's actually a scorpion down there right now. It's on a big vibration-free table. It's surrounded by something called a Faraday cage to eliminate other noise. We've got big manipulators. We've got an oscilloscope. We're going to play it over a loudspeaker. There's lots of fancy amplifiers, and there are computers and so forth. And so the pectin preparation actually looks like this. Here's a scorpion under some clay. We stick a little wire in its tail to complete a circuit. And then we elevate the pectins on a little glass slip, and we put them on a sticky tape. And then let me blow that up. So we're looking now under the microscope, 1 through 21. Let me get really small, down on tooth 19. This is exactly what you would see in my lab as you scroll up and down with our very powerful microscope. Here are those pegs sensilla. Those are the important targets. Let me try to get you even closer still if I could. Here's 19 at the very tip. Those are our targets. That's a peg sensillum, very, very tiny, just about 5 microns across. And we're going to try to put a little wire in the base of one of those and stimulate it then at the same time. And this is what it looks like. I've got a wire in the base of one of those pegs, and I'm going to bring a stimulant very close and actually touch it. And if we're lucky, we're going to hear what this sounds like right now. So what I'm going to play for you is something that very few people have heard. So you're one of the very special crowds that has this chance to hear a sample response from a peg sensillum on the bottom of a scorpion. So this is what it sounds like. And if this is, hopefully we'll get some sound, you're going to see, and I'm going to play this right now. Sound up. Can we get the sound going? You should hear. Okay. Maybe not. Can you play that sound real fast? Let me describe what you're seeing. You're seeing these lines. Those lines are the actual neural impulses of a scorpion. So every one of those lines is actually a nerve cell. Let's play it one more time. I think you'll hear it. We're going to play it over a loudspeaker. There you go. Each of those pops, our ears are really sensitive to this, is a neural impulse called an action potential. In about a couple of seconds, I'm going to blow a 1-second pulse of hexanol, a 6-carbon alcohol, across the top. Here it comes. Right there. See the lines get closer? Hear the pops get faster? That's a neural response. This scorpion is responsive to that chemical. So and now it's going back to baseline. And your neurons work exactly the same way. So I could do the same thing with you if you're willing to do this. So that is a scorpion response. We've done a lot of these over the years. They respond to lots of things: ketones, esters, alcohols, aldehydes. But the question is, do they all respond the same or not? Well, that's a good question. Are they all the same, or are they all different? Well, I had a student named Elise that came along, just an undergraduate, and did a really cool thing. She flooded the pectins with oil. And she said, let's put the electrode in the bore of the recording electrode. Let's put the stimulant in there too. Let's just drop it down and see if we can get a response. And it works. And now she's able to look from peg to peg to peg to peg to see, are they the same or are they different? And this is her hypothesis. So one model might be they're all the same. They respond, all the pegs respond equally to all things. Maybe they're distributed across the teeth, or maybe they're all scattered. Well, this is a very detailed slide, and I won't even pretend to try to go through it with you. What you're seeing are hundreds of records. Every one of these little dots is one of those pops you just heard. And we use citric acid, ethanol, and potassium chloride, a male and a female, and 3 different teeth. The punchline is they respond basically the same. So this is really the model that we're following. So what's good is that we got a whole bunch, tens of thousands of little units that basically respond to the same. My boss, Phil Brownell, actually did an interesting study in the brain where he opened up the brain, and he found that you can find the teeth represented in the brain. You can count tooth 1 through 21 and find it right up there where it empties into the brain. So what we think is going on is it's a really neat tracking system, whereby if there is a female that has put down some sort of a chemical trail, these pectins are set up to almost reflexively guide this animal to the female. And in the bigger picture, of course, this is the answer. Here's the male trying to track a female. He's coming up the trail, and if she is receptive, she will allow him to grab her pedipalps, and they engage in the promenade à deux. And First Officer Spock says the Mojave Desert is indeed a strange world where elaborate tummy tongues help males find females, and where cannibalistic tendencies are somehow squelched by compulsive dances. And I want to thank you so much for being a great audience. All right, I think we might have some time for questions either from the audience here or across the line.
Why don't you stay right there in front of the camera?
Okay.
I'll think if there's any questions. Nobody?
Yeah.
Right here.
How long did it take y'all really to gather up all the research and back
Oh boy, a long time. I really started in earnest in the late 80s. We went over to Germany and figured out how they were doing it. They were actually recording from cockroaches, the antennae of cockroaches, and we learned their techniques and we brought them back, and I started in grad school back in the early 90s. It's been a long time. Yeah? That's a great question. With one of these, the black light, we walk out on the sand at night with our black light, and they fluoresce this beautiful green. It's one of the best things you'll ever do. You really need to do. Ask your mom and dad to get you one of these. Go out to West Texas, all right? Watch out for the snakes. They don't fluoresce. You see, that's what the white light's for. Okay. Oh, doggone it. Well, we got some scorpions in Ohio today, so that's kind of cool. Come see me afterwards. Yeah. Yeah, at night, you cannot see them at all. And so this is a neat trick. We're just lucky as humans that we can see this quite well. But the scorpion, and by the way, this is still an open question. No one really knows why they fluoresce. So I hope one of you, you students out here might get an urge to figure that one out. It's stumped people for ages.
All right, thanks a lot.
Yeah.
We're going to move on to the next
Good,
speaker.
thanks. Thanks so much.
Let's thank him with a round of applause. Thank you very much. All right, we are going to reset here for a moment. Set. Okay. Thanks a lot, Doug. That was great. I love the glowing scorpion. Our next speaker this evening is Cara Shillington from the Department of Biology at Eastern Michigan University, and she's going to talk to us a little bit about some tarantula tracking. All right, I will turn it over to her. Thank you, Cara.
Thank you so much, and it's great to see everybody here. So today I'm going to be talking about some of the work that has been done in my research lab, where I mostly work with tarantulas. And I'm going to be using photographs from multiple different species, multiple different studies. I've done work both in the lab and in the field, so you're going to be seeing many different species and different habitats. But the main theme here that we're looking at is males. And during the mating season, as similar to what Doug talked about, we see these male tarantulas out and about wandering and looking for receptive females. And one of the things that we've been able to do is actually track them with these radio tags, which we see here. And I'm going to talk to you a little bit about what we've been able to learn by following these animals with these radio tags. So I've been interested in tarantulas for many years. They are a big, dominant invertebrate predator. Many people are familiar with this group in general. There is a huge pet trade. There are a lot of people that are captive breeding these animals, and yet despite this, very little is known about these animals in their natural environment. And so this is something that has been of great interest to me. So what I'm going to do today is fill you in on some of those details about what is happening with these animals in their natural environment. And so starting off here, this is a female tarantula. You see here she has a big egg sac which she is guarding. These animals typically are living in burrows, but obviously we chased her out of her burrow for this photo here. She will guard that egg sac. She will protect it. She will roll it over and rotate it to help with the development of those eggs. And she will also regulate the temperature by moving up and down within her burrow to help with the development of those tarantulas. Once again, we're going to do the obligatory photo of the babies as they emerge out of their egg sac. For those of you that know tarantulas, this is coming back to the lab. This was my first and only egg sac of the Goliath bird-eater tarantulas. These are very big tarantulas, and actually they have very, very big babies, but I will echo Doug and say, gee, aren't they really cute? The interesting thing here, so what you saw in that first photo were these are animals that had just emerged out of the egg sac. They would be within the female's burrow. And this is about a month later, and you will notice that there is a lot of darkening of that. And what is happening here is these are arthropods, these are animals that have an external skeleton that have to molt that skeleton in order to grow. And these animals, that darkening is that indication that they are getting close to that time of molting, and they're separating from the new exoskeleton that is developing. And here is what, once they have molted, this is what the tarantulas look like. And so at this stage, these are animals that are now ready to feed. When they are in this stage, they have these really big abdomens and they're full of yolk, and they're actually not feeding, they're using that yolk as nutrients. And once they reach this stage, now they will actually start to feed. In the field, this is the stage that I have typically seen. These animals start to come out of the female's burrow. Here is her burrow here, and all of these little babies around here are actually actively foraging. If there is any sort of disturbance, they will quickly run back into the female's burrow and then come out when that disturbance has moved on. Something that was really interesting was one night we actually saw the emergence of these babies as they moved away from that female burrow. So at this stage, they are now off on their own. And what was really surprising about it was these animals left all in a line, following one behind each other. And we don't have a great explanation for why this is occurring, but this has been seen multiple times. And so I'll show you a little video that I had of this. So you'll see them start to come. So here they're all coming from this side. And now watch, this little guy suddenly decides to take off on his own. So at this point, we now have some going off in this direction, some going off in this direction. So I'm sitting here with my camera trying to decide which way I'm going to go. And eventually, after a point, it just became difficult to follow, and they whittled down to smaller and smaller. But this was them leaving that female's burrow. Okay. The other thing that is remarkable about these animals is the age at adulthood. So for North American tarantulas, males reach sexual maturity at 5 years of age, and females approximately 8 years of age. So from that stage where we see them leaving the female burrow, they set up their own burrows, and they live for 5 to 8 years until they reach sexual maturity. Once they reach sexual maturity, in the males and females, we see secondary sexual characteristics. And these are characteristics that are not directly related to the reproductive systems, but are associated with acquiring mates and passing on sperm. So some examples of secondary sexual characteristics are facial hair in men, different types of antler in deer and antelope, the bright coloration that we see in male birds, and in the male tarantulas, which makes them very easy to distinguish from these females, we see these large hooks on the front pairs of legs. And so when you see this characteristic, you know automatically that this is a male tarantula and that he is sexually mature. And at this point, these males leave their burrows where they've been living for 5 years and suddenly are out wandering looking for females. Another difference to look at, and again, note this is one of my grad students that is holding these. These are wonderful animals to work with. Clearly, they'll sit on your hand for as long as you want. This is the female here. They tend to be much larger bodied. This female has a big abdomen. The bigger the abdomen, the more eggs that she can have, whereas the male tends to have a smaller abdomen and slightly longer legs, which allows him to move about and be very active in searching for those females. Looking at the weight, so essentially what I said with the females, the average weight was 14, the males were 7, so essentially approximately half the size of those females in weight. But the other thing I want you to focus on here is looking at the number of animals. So these are animals that I'm finding out in the field. Essentially, I'm road cruising because it's very easy to find them. You walk along the road and you wait to see where the tarantulas are coming out. If you live in areas where there are tarantulas, you often hear people saying, it's time for the migration of tarantulas. And it's not actually migration, it is the males out walking and looking for females. Note that there are some females, so we are sometimes finding females. I'm not exactly sure why they're leaving their burrows, but we do sometimes find them. But most of what we are finding are these males. So this is where they start searching for the girlfriend. And the males can spend a fair amount of time out there. The interesting thing is that we don't actually know how they are finding those females. Again, we think there are probably some sort of chemical cues once they get in close proximity to these females, also possibly some vibratory cues, but these males are sometimes walking long distances and we don't know exactly how they are honing in on where these females can be found. The next thing, so he does find a girl. What happens then? Does he actually get the chance to mate with her? We don't know. So these are some of the questions that I have looked at with radio tagging these males and following them around. So specifically, I'm looking at the times that they are active. Typically, you hear that tarantulas are active at night, but I want to see what are the distances that they are traveling when they're looking for these females. Are they actually finding females? And I've been lucky enough to work in a couple of different areas. I've done some radio tagging in both Texas and Mexico with different species and different habitats. So I can look at some comparisons between these species to see how things may be different in different environments with different species. So the first thing you do, attach the radio tag. Really easy to do. Nowadays they have very small radio tags, a little bit of super glue, put it on them. Essentially this is just, he's immobilized on a sponge with some rubber bands, just holds him still so that I can get that tag on there for long enough for it to dry. Once again, you can see how easy they are to work with. Not restraining him, put him on the scale, see how big he is now with the tag attached, and then we take them back out and release them where they are found. And we have this antenna and a receiving unit, and each of these tags is on a different frequency, so you can track these individual males. So one of the things we do is we look throughout the night, check at different times of the night to see when these animals are active. And again, a fair amount of variation. Temperature can influence when they are active. They don't like the rain, so you get to sleep in if it's a rainy night. If it's very hot and dry, when it starts to get sunny again, they pretty quickly go to bed. So essentially, you're following these animals around for as long as they are active. So I really like the ones that go to bed early. Typically what happens when they are done with that activity, they find some sort of retreat. So they are looking for some place where they can shelter for the day until they become active the next night. And so here is a little male in here. This is his antennae. He spent the daytime there and then was active again the next morning. In terms of distances traveled, a couple of different things that we looked at. We looked at both the rate, so how many meters are they moving per day, and then we also looked at what is the total distance that they traveled. And so for today, I'm just going to be looking at the distance traveled, total distance traveled. And so one of the things, I'll point out here, first of all, this is a Mexican species, and I'm going to make a comparison with the species that I looked at in Texas, and so you'll see some of the differences there. And so looking at this graph, one of the things you'll notice that the different colors of the bars, and so immediately you can see some of the problems that I was having and some of the problems that these animals were happening. Many of the bright blue bars, those are the males that were being eaten. So put a tag on them within a very short period of time, something was eating them. I had a few problems with equipment failures. Sometimes it was fixable, sometimes it was not. I had one animal that was stepped on by a horse, and then a few animals that were alive. One animal that once I released him went under a big pile of rocks and decided not to move for the entire time period. So a lot of variation in terms of what was happening with these animals, but clearly this is a very tough time. When these animals become out and active, they are very obvious to the predators. And the predators in Mexico, approximately 75% of these were possums. And typically what was happening was in the habitat where I was working, these males were moving out of very dense vegetation into areas of sparse vegetation, and this is one that was passing across a dry riverbed. And I was actually tracking him at the time, and I heard a slight tweak in the beep. 2 seconds later, eye shine from the possum. Possum was actually holding the remains of the tarantula. He dropped them, took off running. The nice thing about the possums, they took off the radio tags, so they never ate my radio tags, so I could reuse them. My radio tags are really expensive. And this is an example of the vegetation that I'm talking about. So in this type of vegetation, they are so obvious. And this is even at night. So these possums, these are nocturnal predators. Possums would pick them up so quickly. You saw I put them out a couple of days, they're being eaten. If they stayed in this dense vegetation, and just for scale here, this is a German Shepherd in that vegetation. Of course, I also have to tromp through this and try and find that little tarantula. And my great helper, German Shepherd there, out with me every single night. And he knew I was looking for the beeps. So every time I tried to find the tarantula, he would focus in on the beep too. So I had to try and look in one direction to distract him while I'm trying to find my tarantula. It was really fun working, but I did have companionship at night. Are they locating females? I'm not 100% sure because I'm not seeing them all the time. Of this group in Mexico, 24% of the animals that I was watching, I did see them find females during the times that I saw it, but beyond that, I don't know for certain. This is a little bit of a cheat. This was one where I introduced the male to the female. So now a comparison of the same type of thing. We did some radio tagging in Texas as well. My average distance traveled was approximately 120 meters. Look at these males in Texas. Huge difference. Different species, different habitat. They were active for a lot longer periods. There's also a big difference between the 2 different years. Are the females maybe more dispersed? They're having to walk further? I don't know, but it was really interesting. Other thing, they're dealing with the same problems. Mortality is a big issue, but what was killing them was different. This little guy is dead. We're not exactly sure what ate his legs off, but at this point, he is dead. The biggest problem here, in comparison, were these tarantula hawk wasps. These are parasitoids. These are female wasps that grab a tarantula, sting it to paralyze it, and then lay a single egg on it. The difference here is between the 2 years, we saw a lot of difference as well. So again, looking at some of the causes of mortality here, once again, we see some problems with equipment. The Pepsis wasps were a big problem. Some went missing. We just didn't know what happened with them. But it was interesting to compare these 2 different environments. This work, a lot of it was done by my student, and I was very glad not to be there because the Pepsis wasps would take and bury these tarantulas with that very expensive radio tag, and this is what my student had to dig through to retrieve them. And here is the little tarantula. Sometimes they could be retrieved alive, and then you had to nurse them back to health. So all of this time, these males are risking life and limb looking for these females. So what exactly are the females doing during this time? Sitting at home. Lots of variation in terms of the burrows, but they are essentially waiting for those males to come to them, and they are thought to remain within those burrows for their entire lifespan. And so finally, just a quick plug for one of my students that is not able to be there. All of the work that I've done so far has been looking at tarantulas that are living in burrows in the ground. And I currently have a student in Puerto Rico that is doing similar types of studies, and we're hoping that the males will come out, but he is watching females that are living in trees. And so here we have an adult female here. This is the little baby that are active, and we're looking at times of activity and looking at differences with male movement when they're now having to go up and down trees. So hopefully he will have some interesting information for us within the next couple of years. That I'd like to thank various people that have contributed to these projects, and I would be happy to take any questions.
Thank you very much.
You're welcome.
We have time for about 2 questions. Go ahead, right here.
This is, Oh, so the question was with the wasp, why are these wasps laying that egg? This is essentially how they are reproducing. So that egg, they will take and bury that tarantula with the egg. The egg will hatch, and the larvae needs fresh food. And the tarantula is that fresh food, so it's paralyzed, so that larvae needs that nutrient. Those are the nutrients for the developing. So essentially, every time I am seeing a Pepsis wasp, I'm looking at that as, was a tarantula.
All right, we need one more. I saw a hand right down here in the front, the pink shirt.
So, you know, these wasps are really interesting, and there are a lot of different wasps. So there are a lot of wasps that are spider specialists, but each one actually has a particular prey. So they target specific prey. So different types of spiders, they target different types of things. And actually, sometimes we've actually found spiders that have been picked up by wasps that we didn't find in their natural environment, but we find them in wasps' nests. Most of these ones, even though they're really big, they're not a problem for us, and they typically ignore us because they really are looking for those tarantulas.
All right, and how about another round of applause for Cara?
Aren't those little tarantulas cute? Our next speaker this evening is Dr. Robert Suter from the Department of Biology at Vassar College, who's going to talk to us about some of the sounds we may not really be able to hear. Bob?
Thank you. Thank you. Hi, everyone. Try to get the technology working here. So I'm going to do two things, I hope. One is I want to tell you something about the technology that we use to make things that are invisible visible and make things that we can't hear audible. And we'll see whether we can make all that happen. At the same time, I want to show you some pretty cool biology that spiders do. So let's get going. This is a fishing spider. It stands on the water surface with ease. It doesn't get wet, and it doesn't sink through the surface of the water. So it can run around on the water surface. Not only that, it can catch small fish and insects that are near it underwater or on the water surface, and it can run and jump and gallop on the water surface. So in what I'm about to show you, down on the right is a Dolomedes, that's the name of the fishing spider, and it's just one jump that I've got in a loop so that you can see it many times. It's jumping on the water surface. I'm videotaping it at 30 frames a second, which is kind of the standard rate that you would use if you were using a video camera. And what you can see is that it happens very fast, and you can't see any of the details of what it's doing. You can just see that it's jumping. If, on the other hand, you want to watch one that we've taped not at 30 frames a second, but at 300 frames a second, you get to see some detail there. Because we've taped it at 300 frames a second, and we're watching it at 30 frames a second. So it's about a tenth as fast as it normally is. Do you want to see it again? This could go on a long time because it's even better the third time, but we won't. So why, you might ask, would these spiders jump vertically from the water surface, aside from just getting stronger and doing calisthenics and all that, and they don't do that? Well, here's the answer. The creature on the right, that guy is a bullfrog. The creature on the left, that guy is, of course, one of these fishing spiders. And this, I'll show you this a couple of times. This is high speed, so it happens very fast, but the taping was done at 300 frames a second. Oh, that didn't work. Let's try this again. So the frog lunges with its mouth wide open. The spider jumps, the frog misses, and curses, or whatever frogs do when they miss. So we can learn all kinds of things by being able to slow down from real time these things that are happening. Why are you still looking at that? Do you still like it? Well, we must go on. So here's what we've just done. We've just taken an event, which I'm representing with this deep red thing. It's a complicated event, takes some amount of time, and we've videotaped it at 30 frames a second. Well, if that's a very short event, then each frame represented by one of these lines, 1, 2, 3, 4, that event is going to be captured by only 3 frames, which is going to go by in 1/10 of a second, and you're not going to get any detail out of what's going on. So what we do to do this is do 300 frames a second or 600 or 1,000 frames a second, so that you get a lot of detail in that time that the event is happening. That's what I've just showed you. So here's another example. This is a spitting spider. Now, just to be a little bit crude about this, you know how humans spit, right? We go, and if you aren't familiar with that, it's because you don't have kids or because you don't watch baseball. If you ever want to see somebody spit, baseballers spit all the time. And when they spit, they put out this smush, right? Not classy. What spitting spiders do in 30 milliseconds, which is a very small fraction of a second, it's about the amount of time that it takes you to blink your eyes once. This is the spitting spider. It's about the size of a grain of rice. It spits a very organized mixture of silk and glue. There's one zigzag here and another zigzag here. And it does that when it's either told to by me or when it's about to eat something or trying to. So I'll show you in real time. So this is 30 frames a second. There's a spitting spider here. There's another small unfortunate spider there, which has already been spat upon once. But when I play this for you, you'll see it get spat upon 3 more times. And what you'll see is the accumulation of more and more silk and glue on this spider that's about to get eaten. One, two, three. Did you get it? It's going to happen again. 1, 2, 3. So it spat 3 times. You got no details about how it did it. All you could see was that it happened because you can see the debris that's the sticky goo that's all over the spider that's about to get eaten. Now, there are other ways of looking at this, obviously. This is a side view of one of those spitting spiders. It's lying on its back so that it's the front of it, the front of the spider is right here. It has jaws there, or chelicerae, and the tip of its jaws have fangs, just like all spiders do. And what happens with these guys is as they spit, they raise their jaws like that while they're spitting, and they flex their fangs at the same time at about 1,000 times a second. So the motion is, all of that happens in 30 milliseconds, and they're spitting through a tiny hole in the upper part of each fang. So that's a lot to accomplish in a small length of time. So this is at 300 frames a second. The spider is facing these 2 wires, which are hanging in front of it, and you'll see the spit come out. It'll wrap around those wires and throw them to the left, and then in a moment of comic relief, you'll see them come back and hit it in the face. Maybe. So that's probably worth seeing again, because one thing to pay attention to is, I told you where the jaws are and where the fangs are. You'll see as I show it again that the jaws do move the way I said they do, but you can't see what the fangs are doing at all. Okay, so here is at 10,000 frames a second, for which you have to go down to the University of Mississippi, where they have the National Center for Physical Acoustics, and they have a camera that can take 10,000 frames a second. I know you want to see that again too. So to orient you, this is the fang right there, a little tiny thing there, and then there's another one right there. You'll see them clearly as it moves them. These are the jaws. The spider is sort of on its side, so it's a little disorienting. This is the face of the spider right here. You okay on orientation? Fangs going back and forth, silk coming out of them, and the fangs are moving at about 1,000 times a second, which I think is kind of cool. And spiders aren't supposed to be able to do that. So, what I've just been telling you also works with sound. And sound is much easier to do because we can record sound at 44,000 cycles per second, and we only care, you and I, about things that go up to about 12,000 cycles per second. And if you're my age, you only care about things that go up to maybe 5,000 cycles per second. And you kids can probably hear up to 20,000 cycles per second, but still, if I can record at 44, I can just slow that down. So we do a recording at 44,000 cycles per second, and this is the graph that you get from a spitting spider when it spits. Nothing happens, and then it spits, and lo and behold, that makes a sound because it's doing a vibration as it's moving those fangs back and forth, so it's going to make a sound. So I recorded from a spitting spider's head, and that's the ray trace, the sound trace, the electrical stuff, and this is a sonogram, and this is what it sounds like. This is in normal speed. You hear it? Okay, so that doesn't give you, right, because it's happening so fast. So we just stretch it out a little bit and play it at a slower rate, which you'll still be able to hear. Okay, and so it's starting at a little higher frequency, dropping down some as it goes. Both fangs are moving, so it's going. How did we get there? Oh, well, anyway, to change slightly what we're talking about, the guy on the left, which is actually a gal, is a large wolf spider, about 3/4 of an inch long, the body, something like that. And her baby is right there. And you all know what a match looks like. So that baby is really tiny. And when wolf spider babies are ready to go somewhere else, having had enough with living with their parents, actually with just one parent, they often climb to the top of a stem or a stick or a piece of brush. If it's a slightly breezy day, they will stand up on tiptoes, stick their rear end in the air, let a little silk out, and the breeze lifts them off there and they fly away. It's called ballooning. Lots and lots of spiders do it in the spring and summer. And so I'll show you what that looks like. This is at real time. Standing on tiptoes, getting more, gone. But gone very fast, right? That guy went so fast, you couldn't really see very much in detail. I'm not going to show that one again. The second one is, again, at high speed, and this one's, you'll see some details. For example, that it actually jumps off. It doesn't just get lifted off. Ready? Cock its legs, jump. Okay, that's one set of processes that happen very fast, and we have to slow them down. Other things, scientists don't like to be bored. Some things happen for a really long time, and you have to speed them up. So instead of doing it at 30 frames a second, maybe we do a video of this long process at 1 frame a second. This is how they cut down the trees near our house, right? So the process took about 4 hours, and at a frame a second, you got to see it in, you know, whatever that was, 10 seconds. Now, might we use that for? The spiders that spin these really cool, or bend these leaves, which are cattail leaves, and make capsules in which they live and in which they lay their eggs and hatch their babies. They do that. It takes them about 6 or 8 hours to make one of these capsules. Here's what it looks like at 1 frame per second. Boring after you've seen it for a little while. And I've already cut out the guts of some of these, so you're just seeing parts of it. And it bends the leaf, and it's going to bend it all the way eventually. But I'm even bored at looking at the videotape. So that allows me to get, in the course of a 6-hour recording, I don't have to look at all 6 hours. I can just take data points from once a second and make graphs like these that tell me something about how this actually happens. I'm hurrying along because I've been warned I don't have very much time. Another thing that happens that we worry about is there are lots of events that happen like this. There's something happens, nothing happens, something, nothing, something, nothing. So you use security software. Security spy is what I call it. It's surveillance stuff. Same kind of thing the NSA uses, though theirs is better. And so you, instead of doing that kind recording the whole time, you just record the parts that you're interested in, and software takes care of that. And so here's a spider that's digging a hole. It's a geolycosa, a burrowing wolf spider. You're seeing about 2 hours of behavior compressed. Not that the behavior is any different than you would see, it's just that we've gotten rid of all the spaces that you didn't want to watch. Right now it's scraped the stuff, and now it's making a little ball of what it scraped as it makes this burrow. And in a moment it's going to turn toward us and throw that chunk of sand and dirt. Phew, gone. And I only had to spend about 5 minutes looking at it instead of 3 hours. Okay, the last thing I want to show you, I think, is this is a brown recluse spider looking right at you. And in real time, stimulated the right way, it moves its pedipalps very fast next to its jaws, and it has on its pedipalps a peg, which is this thing right here, and a file, which is this thing right here, and it rubs the two together. And this is what it sounds like. Now, how cool is that? I have to tell you that the technology that allows this to happen, I could do it with a laser vibrometer, laser Doppler vibrometer, which would cost me 10,000 bucks, or I could do it with this, which is a phonograph needle, and I get the same fidelity, it turns out. And ask your grandparents what a phonograph needle is. Thank you very much.
We've got time for a couple of questions, really quick. Anybody? Okay, I don't see any hands.
Okay,
well,
thanks.
Thank you very much, Bob.
All right. Now I will call to the stage our next act. Doctors George Uetz and Dave Clark. George hails from the University of Cincinnati, Department of Biological Sciences. Dave Clark comes from the Department of Biology at Alma College up in Michigan. They're going to talk to us about a little bit of virtual sex.
Are we now?
Or virtual dating, I suppose.
Or something.
All right.
Thanks, George.
Well, what Dave and I want to share with you tonight is some of our research with digital video imaging and how we use that to try to understand how animals communicate. Now, spiders have 8 eyes usually, some of them have 6, but their eyes are not really great. Spider vision is pretty poor. A whole lot of spiders can only see light and dark and moving shadows, but there are some spiders whose vision is really good. And among them, the wolf spiders up here and the jumping spiders over here have really good eyes that in many ways are as good as our own. And an example of this is the jumping spider eye. The jumping spiders have a set of eyes that they use to create a panoramic vision. They see behind them as well as in front of them. They see above them as well as in front of them. But when one of these eyes detects motion, the spider swivels and focuses his incredible anterior median eyes, which we see in cross-section here, these big headlights up here, and is able to see high-resolution images. And on account of this, jumping spiders, who also, by the way, see color, have communication systems where the males make visual signals towards the females, like this peacock spider is doing. And a whole lot of the behavior of jumping spiders is visual mediated behavior. And spider eyes and human eyes are very different. We have good vision, as do many vertebrate animals, but our vision is often directed straight ahead. We have peripheral vision, but we can't see out the back of our head. Spiders, on the other hand, like I said, they have 8 eyes and they can see in this large panoramic view. They also see different colors than we do. But there's one thing that we have in common with them, or they have in common with us, and that's the ability to see moving images that are projected as a rapid series of still images that we call film, or nowadays video. And so they have a same feature of their eye that enables them to see TV, and that's pretty weird, don't you think? Dave and I had discovered this quite some time ago, and you can tell by this National Geographic photo that it was quite some time ago. But what we found is that spiders respond to video images on microscreen TVs, and this gave us the opportunity to do something that we had never been able to do before, which is take the same image, the same movie, literally, of the spider doing natural behavior and show it to multiple spiders and see their responses. And at the same time, we could capture that video, we could modify it in some way, change the color, change the behavior, change the speed, and look at how what they see gives us some insights to the inside of their communication system. So Dave is going to explain this a little better.
Great. Well, thank you, George. It's a pleasure to be here. My name's Dave. To say that the technology has changed since the time that we discovered this would be quite an understatement. When this was actually discovered, I was using 8mm film at that particular time to do this, and the video that we used to use was what was called analog video. And many of you probably remember that back at around this time, a large TV screen would have been like a 27-inch CRT, that sort of thing, or a cathode ray tube type of television. And they all used what was called this analog video. And analog video has a relatively limited ability to manipulate. One of the easier things that you can do with it is what's called chroma keying, or placing an object against a green screen or a blue screen, and then easily change the background against which that object is placed. You can make people look like they're flying through space or flying through the air like this, and some of the fancy special effects that they used to use back in that time. But things have changed dramatically with digital video, and with digital video, many different possibilities exist. And we sort of switched over to that, started using computers to generate computer animations of the various stimuli that we were showing to spiders. And so we moved away from this whole analog kind of video stimuli and moved into the realm of digital video stimuli. And with that, you can easily manipulate the behavior of the stimulus. You can even change the morphology up and make it look completely different. And those of you that are gamers out there know certainly the potential power of interactive digital video. What you see over here on the right-hand side of the slide, I think, is just a tremendous example of the use of 3D digital video sorts of stimuli. And the ability to do this today is incredibly easy. In fact, people can even manipulate video quite easily on things like iPhones or iPads very easily. If you want to get a little bit fancier, the sorts of things that you might need to do this would be a decent video camera, a laptop computer, and some memory to store these, because video is still pretty memory intensive. But the video editing software and the animation programs that allow one to do this sort of thing are relatively easy to use these days. And I want to tell you a little bit about how we've actually used this, and I'm going to tell you one story here, and George is going to talk about another one. I'm going to talk a little bit about a spider that's near and dear to my heart. This is called the dimorphic jumping spider, Mavian clemens. And this is a really strange species of jumping spider where there are two different male morphs in this particular species. The tufted morph that you see up here on the top has this all black body. It has these three tufts above its eyes, like you see there. Its pedipalps are this jet black color, and its legs are all iridescent or kind of a pearly white color. In contrast, the gray morph is this zebra stripe thing that you see down here. Now, not only do these males look different, they also behave differently when they court the female. The tufted male stilts from a stationary position, often at a considerable distance from the female. The gray male actually does this sort of sidling back and forth behavior that you see really pretty close to the female. And one of the questions that we asked was, how do females recognize two different male types? And this is where video imagery and the use of digital animations really allowed us to probe into the question about the female recognition of these two different types of males. These are some of the actual videos that we used. This is a tufted male displaying really a quite accurate phase 1 courtship behavior as if it was from the perspective of the female. This is the gray male down here performing its behavior from the perspective of the female. These were shown to them on little tiny video screens. And we could do things such as manipulate the morphology and the behavior and then see which it is that the females responded to. As it turns out, for the control males, females respond about equally to these males that look and behave like themselves. However, if you manipulate them and make a gray male, this guy down here, behave like a tufted, it decreases the female receptivity response to about 40%. But if you make a tufted male behave like this guy, females don't like him at all, and only about 3% of the females showed any kind of receptivity to it. In fact, they tended to attack that male. This suggested to us that for the tufted males, behavior was more important, and for the gray males, the morphology was more important. And some of the things that we've been getting into is the use of 3D technology to try and address some different questions. This has particularly been useful for asking questions about what it looks like from a predator's perspective, using this image, turning it around like you see here. And what's interesting, I think, is that the surface area that's exposed by a tufted male is the same whether he's looking at you or whether you're looking at him from the upward in the vegetation. That's not true for the gray male. If you look at him from the front, he looks like a small little dot. However, if you're a bird or you're a wasp that's patrolling in the vegetation, up there, he really exposes quite a bit of area to them and is potentially at risk of being spotted by these potential predators. Well, George is going to come back and tell you about some of the wolf spider work that we've been working on.
Thanks, Dave. One of the wolf spiders that's very common here in Ohio, the brush-legged wolf spider, Schizocosa ocreata, is a very interesting spider because it has very complex courtship behavior. Now, females are very choosy and males are not. Males try to mate with as many females as possible in the spring during the mating season, but females can only mate once, and as a consequence, they're very choosy. And this puts sexual selection pressure on males to be effective communicators. And these males have complex courtship that involves visual signals. They have these decorated legs with tufts of bristles on their legs. They wave them in displays, and they can be seen at some distance in the leaf litter. And they also produce vibration by percussion and by special stridulatory organs that they communicate with the female. So this is a complex courtship behavior, and we have a lot of questions about how this kind of behavior might evolve. One of the prevailing theories about complex courtship behavior, of course, is, to put it in the vernacular, the chicks dig it. The more complex the male's behavior, the more showy a male is, the more likely he is to get a date. And so we were asking the question about these tufts of bristles on the male's leg. And we noticed that if you look at live males, females tend to prefer the ones with bigger tufts, and not so much for the ones with small tufts or ones that we've shaved off. But what we couldn't tell is whether this was also just a function of something else, like the male's size or the male's behavior. Maybe the guys with the little tufts are kind of weak in the display. And so what we were able to do with video is take this rendered male and go in frame by frame and shave his tufts off, make them smaller, or even make them larger. And we're only changing one feature, but everything else is exactly the same. And as you can see, some of you are old enough to remember the commercial, is it live or is it Memorex? But there's no difference, no statistically significant difference between the video playback and the live male. And so we verify that it's the tufts that females are really keying in on when males are communicating to them. That said, these animals live in a very complex environment, and male signals travel through leaf litter to varying distances, but this is the range, this is a female's sensory range. This is as far as she can detect his signals, and that's no trivial matter, because if the message gets through, he may get to pass his genes on, or if it doesn't get through very well, he may contribute to her next meal. So you can imagine that natural selection and sexual selection would favor very conspicuous courtship, but there's a price to be paid for that, and that price is that the females are not the only animals that are listening in. We have animals that we call eavesdroppers, like predators and competing males that also perceive these signals, and they may exert some kind of influence on the success of the male. One of them, for example, are these predatory toads, who also respond to video images. And so what we've done here, as you can see, is present a video to a toad. And as you'll see down here, these toads prey upon these spiders quite readily, and they will also prey upon them on a video screen. And by manipulating the image and just taking the tufts off, we change the attack rate of the toad. We can also do something, and this is what the guy who's waving his hand and telling me I'm running out of time, Andy Roberts, our host, did for his doctoral work, was to ask the question, a virtual evolution question. What if the tufts got bigger? Well, we know that tufts, that females prefer big male tufts. Why don't they get bigger and bigger and bigger over evolutionary time? And the answer is that by making them larger and finding out that toads key in on them quicker, that predators put the brakes on that kind of evolution. Another thing we found, also part of Andy's doctoral work, is that we asked the question, well, how do males respond to other males on video? We expected them to attack them, and this was a great way to stage some encounters and see aggressive behavior, but we were surprised to find that they court. And we wondered about that. We were a little concerned, but ultimately what we thought we were seeing something, what we figured out is that males are eavesdropping. They're listening in and paying attention to other male courtship, because if there's another male courting a female, there's a female nearby. And so we've been asking questions about how does a male key in on another male and steal his date. And so we did these experiments where we present the male with a blank screen, and he doesn't do anything. He ignores it. He explores the arena, and then on the screen we put a courting male spider. And all of a sudden he begins to court, and he looks around the arena and he courts. And then we take the image away after another 3 minutes, and he continues to explore and court. In fact, his rate of courtship matches that or exceeds that of the male on the screen, which is a way to even the odds. Our current research involves both sight and sound, and I'm afraid that sound isn't playing very loud right now, but this little device here is called a piezoelectric disc bender, and it's the speaker in your cell phone, and we are able to play back vibrations along with videos, and we can look at what females really key in on males, and while they respond to visual or vibratory seismic cues equally, they respond even more when those are presented together. And so it's this complex courtship that really gets the females interested. So what have we learned from all of this? Well, despite what a lot of people fear, spiders are really great animals for research. And we've developed a novel method that enables us to ask questions about animal communication that a lot of other people are using with a lot of other animals, toads, to lizards, to birds, to fish, to even octopus. And this has given us a lot of insights into the sensory systems of animals and their communication. And it's also taught us that spiders can learn. It's taught us that spider behaviors and their cognitive abilities are a lot more complex than we ever thought. So I hope I have time for a question or two.
Absolutely. Time for a couple questions?
Anybody out there on the internet?
Not so far.
Not so far. Okay.
All right. Well, thank you very much,
Yes.
George, Dave.
Thank you.
Thanks a lot.
Thank you.
Let me get this off of here for you. I'll come back and get my phone later. Okay, I just pull it. Let me close this out first.
All right. Thank you, guys. Up next, Rick Vetter, Mr. Rick Vetter, who comes to us from the Department of Entomology at the University of California, Riverside, who's going to talk to us about misconceptions of the brown recluse.
Actually, misconceptions of the brown recluse in Ohio. No, which one do you want me to use? This? Okay. I've been working on brown recluse for over 20 years. Just about 20 years ago, I realized that there really was no arachnological expert working on this spider, and that mostly the information being put out there was being put out by medical people and people who had no knowledge of the spider itself. And so for 20 years or so, I've been trying to put out some solid information, solid scientific information about the brown recluse and reaching all parts of the country. So pretty much for 20 years, I've had a crusade going on trying to educate folks about the recluse. Now the problem is that people misidentify, the brown recluse people will say, oh, I understand it has a violin. And yes, it has a very nice violin marking, as you can see on the spider. The problem is that people are very creative in finding violins in every single portion of a spider's body. The thing is, what I want to show you is with a recluse, you should not use the violin marking to identify a recluse spider. You should use the eyes. Most spiders have 8 eyes and 2 rows of 4. Brown recluse has 6 eyes, a pair in front and a pair on either side, separated by a space. And but that's what you should use, because here's 6 examples of spiders that are sent to me as brown recluses. None of them are, but you can see the variation that people variation that occurs in spiders that people misidentify as recluse spiders. Now, one of the problems is that people twist things around. They learn one piece of information and then they twist it and get it wrong. For example, if I tell you birds have wings, they go, birds have wings, birds have, if it has wings, it's a bird. And then, so now all of a sudden bees are birds, bats are birds, pterodactyls are birds. And so the same thing happens, that people learn that brown recluses have violins, therefore if it has a violin, it must be a brown recluse. And so I want to show you how you can identify and misidentify a brown recluse. First of all, they have a uniformly colored abdomen. It can vary from a light tan to a dark brown, depending on what it eats. This is the same spider being fed crickets, and then I fed it 2 houseflies, and the abdomen turns black. So you cannot use the abdomen color. But if you see more than one color on the spider's body, that is not a recluse spider. So if you see white dots and black areas, not a recluse. Its legs are covered in fine hairs that lay down real nicely, not spines. If you can see big frickin' spines on a spider, it is not a recluse spider. The legs are uniformly colored. The front legs may be a little bit darker in males, but if you can see lines, if you can see spots, if you can see stripes, or any other 2 colored markings on the legs, it is not a recluse spider. It does not make an exposed web. I get people telling me, oh, I went outside my backyard and I ran into a recluse web stretched between 2 trees. No, they make a web behind the bookshelf or a painting or something. If you can see a spider in a web, it's not a recluse. And they're only about 3/8 of an inch long, and people equate large spiders with dangerous. So if you've got the small spider or a big spider running through your house, it's not a recluse. Now, the brown recluse in Ohio, this is one of the areas, I just want to go through this, because people believe there are brown recluses everywhere through North America, causing all kinds of damage, decapitations, the whole thing. Now, so take your typical person from Ohio. Do you recognize yourself in there? Of course you do. Okay, well, the arachnologists say, well, there are not many brown recluses in Ohio, but they say, oh no, you're wrong. You don't know what you're talking about, because I saw this brown recluse making a funnel in my backyard. I saw this brown recluse over by my barbecue. This brown recluse is running around my barn. I saw this brown recluse running across my kitchen floor. This brown recluse is in my cellar. I saw this brown recluse over by the patio. I saw this brown recluse by my fishing boat. I saw this brown recluse when I went to a farm. They're all over the place. Okay, this is where you find the recluse spider. It's found from Nebraska down to Texas. It pokes its way into Ohio, into northern Georgia. It's known as Loxosceles reclusa, and it does get into portions of the Midwest here. Well, I did a study. I said, if you think you have a brown recluse, you send it to me, and I will identify it for you. I had over almost 1,800 spiders sent to me from 49 states, including 12 from Alaska, people thinking they had recluses. Now, let's get rid of the non-recluse spiders here and watch what happens in Ohio. They disappear, and all of a sudden, you've only got 1 spot in Ohio in the study that had recluses. Let's go. Of the Ohio submissions, 33 spiders sent in, 6 brown recluses from 1 barn in New Lebanon, 27 other spiders from different families. We had 3 on top there, 3 funnel web spiders. 7 of the woodlouse spider. It has large fangs. It moves them out, and they're very intimidating. So people think large fangs must be dangerous, must be a recluse. The bottom here is a barn funnel weaver. The one here is over the far side is a wolf spider. It can see you. It runs away when you come running or turn on the light. So if it runs away, it must be doing something bad. It must be a recluse. The cello spider up top there, it does have a marking on its cephalothorax. It's a lot like a violin, so you get half credit. The one on the bottom here is there's 2 of those. It's a Amrabius ferox. It's a European spider. This big honking spider that you see when you turn over your boat or canoe, that sort of goes across the canoe, makes a lot of noise. That's a fishing spider that Bob Suter talked about. And so I just want to sort of explain, try to make the analogy that the distribution of an animal, or say the brown recluse or any animal, is very similar to that of a rain cloud. If you stand under the middle of a rain cloud, you're going to get wet. There's going to be lots of raindrops, which represent the spiders. Lots of raindrops. There's going to be a real short time between when 2 raindrops hit you on your body. There's going to be a real short distance between those 2 raindrops. As you march outside and go out of the rain cloud, there'll be fewer and fewer drops. And as eventually you get out into you'll be at the edge of the cloud. There'll be the 2 raindrops hitting you. There'll be a big distance between 2 of those drops, and there'll be a big time differential between those 2 drops hitting you. As you get out into the sunshine, no more rain. Well, if you want to find recluses, you go to Tennessee and Kentucky. They're thick there. Lots of those spiders. As you start getting up into Ohio, you're getting to the northern range of the spider. They're starting to disappear. You might be able to find them in Cincinnati. You started getting into the middle of the state, up in the Akron area, they disappear. And now, another thing that's interesting is the center of the American arachnological universe is Ohio. There are arachnologists working in the state. There is no other place that has more arachnologists. In fact, you can't swing a cat in Ohio without hitting an arachnologist. They're all over the freaking place. Okay, now the other thing is that a lot of the work that's being done is the knowledge from the Ohio spider fauna is known by Rich Bradley. He's the one on the right. And Rich Bradley is retired from OSU Marion. He did the Ohio Spider Survey, 20 years of collecting spiders in Ohio. Over 40,000 spiders being collected, 642 species. This is a lot of data. This is a lot of knowledge from Ohio. So how many finds of brown recluses? This is pretty much it. There's only a few been found in houses across the state, not many at all. And some people go look at this and go, look, it's like they're the tip of the iceberg. They're not really the tip of the iceberg. They're more like an ice cube. It melts away, it's gone, it's out of here. And so there really aren't that many brown recluses in Ohio. Now the problem is that physicians will look at a skin lesion and say, oh, well, that's a brown recluse bite. And this happens all over the country. It's not so much anymore, because I'll get into that in a few seconds here. But the problem is doctor will say, well, that's a brown recluse bite. And then the patient comes home, and they want to argue with me or an Ohio arachnologist saying, no, it's like, no, there are brown recluses all over the place. My doctor said, well, no, the doctor's wrong. There aren't many brown recluses around here. But my doctor said, so, well, no, it's wrong. But indeed, your doctor is wrong. A doctor cannot look at a skin lesion and identify it as a recluse bite. There's an example. How about this one? This was published in a medical journal showing what a brown recluse bite was. A physician diagnosed it. No spider was seen in the incident. Published in a medical journal. Winnipeg, Canada. No brown recluse has ever been found north of the southern Canadian border, yet this was published as a brown recluse bite. Is this a brown recluse bite on this guy's leg? Doctor said it was, diagnosed it. Is this Sierra Nevada? Don't worry, we treat them all the time up here. No, you don't. You don't have brown recluses in Sierra Nevada. He tore his house apart looking for spiders, found none. No spiders. You know what he did? He was cleaning his oven wearing shorts. He knelt an oven cleaner. It's a chemical burn. He had a physician diagnosed as a recluse bite. Is this a brown recluse bite below this guy's knee? Doctor said it was. This happened in Rhode Island. Guy was vacationing in Connecticut. He had Lyme disease. I warned him about it. He said his first tests for Lyme disease were inconsequential. The later tests were off the charts for Lyme bacteria. It's transmitted by a tick. Is this a brown recluse bite on this baby's arm? Doctor said it was. New York City, it happened. This was anthrax in 2001. Thank you very much. 2001, where they had the bioterrorism situations. Is this a brown recluse bite? No, it's pyoderma gangrenosum. It's not no known cause, and it's often mistaken for a brown recluse bite. Is this a brown recluse bite? No, this is a pizza. It's got cheese, pepperoni, oregano. Folks, this is serious stuff. They say, well, it's not a brown recluse bite. What could it possibly be? There are a lot of different things that are mistaken for, can be or have been misidentified as a brown recluse bite. You have staphylococcus, streptococcus, anthrax. You've got Lyme disease, herpes simplex, or herpes zoster, shingles. You've got fungal infections that get into landscapers and such. Poison ivy, poison oak, chemical burns. You've got blood diseases, leukemia, lymphoma, basal cell carcinoma. People are having cancer and being told they have spider bites. Diabetic ulcer, bed sores, self-induced injuries. People will say, well, what about that Ohio prisoner who died years ago from a brown recluse bite? Well, a few days later, another article comes out saying, well, prison tattoos, not venomous spiders from these people gouging themselves with contraband needles. But it swelled to the size of a baseball. Well, it's not a brown recluse bite because brown recluse bites are flat. But it oozed lots of pus. Well, that tells me it's not a brown recluse bite either because it's dry. Pus means bacterial infection. But the doctors gave me antibiotics, it got better. Well, that means you were misdiagnosed for having a spider bite, and you were given the wrong treatment because antibiotics do nothing for venom, and they actually kill bacteria. But they gave you the right medication for the condition you had. Now, the thing is, there's a big thing coming out in the last couple of years, MRSA, methicillin-resistant Staphylococcus aureus. Once people, both the general public and the medical community, became aware of MRSA as a condition that causes skin lesions, the contacts I've been getting for brown recluse bites have dropped from about 30 to 50 a year down to 1 or 2 a year. A lot of bacterial infections probably ended up being these MRSA infections, and now that they're being treated as such. So there are a lot of arachnologists in Ohio, and so if you still want to believe that there are brown recluses everywhere, it's actually going to be more like a needle in a haystack. And if you still want to believe there are brown recluses, and you walk up to an arachnologist in Ohio and say there are brown recluses everywhere, don't be surprised if this is the reaction you get. Done. I probably have 15 minutes still. Oh, do I have time? Questions? You're stunned.
Very fast. I'm reeling a little bit. Go ahead, right here.
I have friends in California who tell me they know people who have been bitten by a brown recluse. So many states now.
I'm sorry, there are children present.
I'm from California. I've been working with them for years for there. There are no brown recluses in California. There's a desert recluse in the desert, up in the high desert and low desert Palm Springs area. I've got a publication that shows all the doctor's diagnoses of that. It happens all the time. People will swear that they've been bitten by brown recluses. You go to a doctor and the doctor gives you, oh, well, it could be pyoderma gangrenosum, it could be erythema chronica migrans, it could be bed sore, it could be a spider bite. They give you that. The patient comes home, I was bitten by a brown recluse. That's the only thing that there's a psychological aspect to it. People glom onto this brown recluse bite thing, and you cannot pry it from their white-knuckled fingers. I mean, they want to be bitten by, I survived an attack by a great white shark. I was attacked by a rattlesnake. It's like, I had a bacterial infection. It doesn't happen that way. People do not go around claiming that they, or proud of the fact they had a bacterial infection. Brown recluse bite is a big thing. I've been dealing with this for over 20 years in California.
Gary, is that you back there?
So when people have a legitimate symptom, they go to the physician. How do you recommend they talk to their physician if they want to question it?
Okay, the question was, a person has something to go to the doctor. How do you deal with this situation? What they should be doing is not giving it a cause. To say it's a brown recluse bite says that they know this wound was caused by an animal biting them. What they should just do is treat the wound as for whatever it is. It's a lesion, take care of it. Okay, do I have a picture of what a brown recluse bite is? I actually personally do, but the problem is it varies from like a mosquito bite, the rash that just goes away in a couple days, to a horrible gaping wound. And so it will cross the spectrum of probably 100 different conditions. And people will ask me, why don't you put some pictures on the internet of what a brown recluse bite looks like? It's like, oh, great. Instead of people verifying it, then everything they have is going to be a brown recluse bite. They look at those pictures of those spiders and go, spider looks just like the picture on the internet. It's not even a spider sometimes what they hand in to me. Any more? Moving on?
Okay.
All right.
All right, thanks, Rick.
Whew, that was quick. Okay. Our last speaker of the evening is Joe Warfel, who is a photographer some of the time. Are we focusing? There we go. And Joe takes some spectacular photographs of spiders all over the world, and he's going to share some of what he has taken with us tonight. It's actually probably good that we have a little bit of extra time, because then we can pause a little bit on some of the pictures and take a little time to peruse them. So I'll introduce him now. Come on up and show us what you've got.
Well, welcome. I'm glad to be here tonight, and I have basically a selection of images that I hope to sort of share the whole family of arachnids that most people, you know spiders. You've seen earlier tonight, you've seen little talks with scorpions and some other arachnid organisms. So I'm going to cover at least a few pictures, if not a whole lot of pictures, depends on the order of the various groups that encompass arachnids. And the only one that I won't have, and even some of our arachnologists here probably have never seen one, and I've never seen one, something called a palpigrade. All right, well, so there we are. Top casual. All right, well, we'll get started and start seeing some of the arachnids. Well, arachnids as a group, including the scorpions, spiders, and others, are a very ancient group. And we have some actually relatives, doesn't want to keep, we have ancient relatives still with us that have been around for a few 100 million years. That's horseshoe crabs. They're closer related to spiders than they are to crabs and lobsters. Ancient life, here's a spider trapped in 30-million-year-old amber. This is a sample of a early it's called Lephistheus Malaysian. Can I get this? Sorry. All right, I'm sorry about that. This is a Malaysian trapdoor spider, and it's got a lot of what's considered primitive spider features. There on the abdomen, you can see little scoots that the modern, other spider groups don't have. It's more or less like a leathery bag in most spiders. But these live in little burrows with trapdoors. Very cool. So the next group, you've already heard some about scorpions, and they're another very ancient arachnid order. And here's one that is just hiding at the entrance of its burrow in Arizona. And as Doug showed you earlier this evening, under black light, it stands out very, very well. And so that's really the best way to go when you're out in the desert to look for them. You can buy it at a lot of stores, a little cheap black light flashlight, and you go around and look for them at night. And you'll be amazed at how many you can find, and if you're in the right location. You heard about the mating and the whole courtship dance of scorpions. Well, before I realized what was going on many years ago when I photographed this was late at night on the highway. This is asphalt that they're on. And I thought, oh, they're fighting. Well, this was the courtship or the male positioning the female, so it could pick up the spermatophore that it probably laid down on the asphalt. Just a little close-up of the chelicera, the mouth parts of a scorpion. This is one on a tree trunk out in Costa Rica, in the rainforest. And as some of the other pictures you saw, here's the little babies. This is when the babies are very, very young. They don't have any color. They'll climb up on the mother until they molt at least once or twice. Doug could probably tell you more details about that. So the next group, pseudoscorpions. They look very, very much like a scorpion, but they don't have the telson. They don't have the tail and the stinger. But these things are very, very small. A large one would be 1/4 of an inch or 1/3 of an inch, if that. Most of them are a few millimeters long or less. This one is a specimen I collected in rainforests in Guyana. This one's a different one, but this was on a tree trunk. This one's from Tennessee. This fly was in Massachusetts, and the pseudoscorpions that you see here, they're holding on with their little pinchers, and they'll often grab the antenna of an insect or the leg of the fly, such as here, and they're not eating it, they're not attacking it, they're not doing anything bad other than being a bother. They're getting a hitchhike ride by the fly, and they just hold on, and the fly can't pull them off. They can't knock them off. They try to clean them. No way. It just won't. They hold on so tight. And when they decide that, oh, the fly landed at a nice place, they'll let go and wander off. And it's called phoracy is the behavior. Okay, the next group of arachnids is called vinegaroons. The scientific name, they're called Europigids. This one is one I saw in Guyana, in the rainforest. This one's a fairly small species, 1/2 an inch, 3/4 of an inch long. This is a male and female of Masticobroctus genus in Arizona. This is a male and female, rolled a log over, and they were below the log. And these can get a couple inches long or more. So the next arachnid is common name they call whip spiders, sometimes tailless whip scorpions. Amblypygid is the scientific group that they belong to. And if you saw Fear Factor a few years ago, they had cave spiders that they had to eat. Well, that's what these were. They're not spiders, but they're harmless unless you're an insect. They don't have venom. They don't have a stinger. They just are, they come out at night and they live in crevices or on tree trunks under bark. They're very, very flat. And they just grab their prey with the pedipalps, those big spiny things that you see. The immatures, the young ones, are usually very much more colorful and patterned than the adults. And as they get old and mature, they usually darken and get more bland looking. And this is another immature, as this is with the color, but it turns dark when it reaches adulthood. Here's an adult, and you can see that they don't need venom. Oh, excuse me. If they reach out and grab you and touch someone, that's it. The second pair of legs are modified. See these long legs? They don't really use them for walking. They're like antennas. They search the world, and that's how they really see. They do have eyes, but most of their world is visualized through those legs. And if those legs are lost or removed, they almost are blind. They don't know what to do. This is a male and female out at night in Guyana. The next group, Arachnologids. It's a fairly small number of species in the world, the tropical. This one's from Costa Rica. They have a unique feature that no other arachnid or any other insect have. It's that covering their mouth parts and their chelicerae and their fangs, they have this little item here called a cuculus, but it's like a little hood, and they can retract it somewhat like a garage door and expose their mouth parts when they feed. And this is another one. This one is entomologist friend loaned me this for photography. This species was a newly described species he found and described from the Tiwa rainforest in Ghana, West Africa. Next group is schizomids. I'm sure you've seen a lot of these. No. They're not too commonly seen either, other than by arachnologists searching for them in various tropical climes. And these can be only half an inch long or so, probably averaging, maybe a little bit bigger for some large species. Then another group is called the wind scorpions or wind spiders. Sulfugids, or sulfugids is the scientific name for the group. And the name wind scorpion is very apropos because they run like the wind. They're extremely fast runners, and they're generally in dry habitats and deserts, or fairly xeric habitats. This is in Arizona. He captured a small grasshopper. And they run around with their palps, what look like big legs, sticking kind of right out up front, and they'll just run around a million miles an hour, running around there. This one is in Arizona at night, and it's digging a burrow. There's the burrow there, and it's bulldozing soil, going in every 30 seconds or so, and it'll come back out and shove out the dirt. This one looks gravid, so it may very well be a female with eggs, and I believe they lay them under in the soil. This one I found under a rock. It was daytime, and they hide away generally in the heat of the day. And it was about 100 degrees out, and I rolled the rock over, and you can see how moist and damp it is under there. So even though they're a desert animal, they like the moisture and they like humidity. So they would only be out at night when things are cooler and when the humidity is higher. As fast as they are, I did find this scorpion, desert hairy, that had captured a solifuge and was eating it. Likely just the solifuge was running around 100 miles an hour and probably just have to bumped into the sitting scorpion and just latched onto it, because there's no way I can imagine that the scorpion can run fast enough and catch one of these. Because the next group, harvestmen. Many people call them daddy longlegs. I don't like using that term because there's also a spider, the cellar spider, that common name's called daddy longlegs, so it's a little confusing. Harvestmen relates to the animals being very commonly, particularly in the northern climes and North America and all, they're very common in great numbers later in the season during the harvest time in the fall. And so that's where that name comes from pretty much. And these guys are pretty much all legs, as you can tell, And it's very important for their sensory awareness, and also they're always cleaning their legs. So even their hind legs, as long as they are, they can bend them around through their mouth and clean them and clean the feet. Few species every once in a while will aggregate into little small groups and hang out together. This is a very small aggregation. Sometimes it can be very great numbers. In some caves in certain areas, you can find hundreds or thousands, all bunched in a very small area. This is looking sideways. This was underneath a park picnic table. Now in the tropics, I've generally found them on large tree trunks, and they tend to be less dense, so their aggregations are generally fewer. And often when you approach them, the whole mass of harvestmen will start bouncing up and down on their legs and jiggling and blurring. This is a typical stance of a lot of harvestmen. So you see that second pair of legs, which they do use for walking, but they also use those, like the amblypygids, for sensory awareness of their environment. This is one that is molting, or is in the process of molting. So it's hanging from underneath a downed log, and when it molts, it has to pull all that long leg out of the old skin to get it, and then it rests there until it hardens and dries, and then it can walk around. But it's pretty much helpless until that dries out. This is I rolled a log over one day, and this very tiny harvestman, most of the babies and the small ones will hide under crevices and under rocks and under logs where they're protected and where it's damp and moist until they reach adulthood and they start wandering around. This one's in the process of molting, so he's pulling his legs all the way out to try to, out of the old skin and then legs to stretch out. And then now he's finally got them out, so now he's stretching. And he'll have to dry out for a little bit before he's able to walk around. This is just a colorful sample of some in the tropics. These were from Peru in the Amazon. This was in Chiapas, Mexico. The legs span here probably about 4 or 5 inches, maybe more. There are groups belong to a group called Lanatores, that the harvestmen, instead of their very long legs, they're very chunky, short-legged and small. The body on this is probably about 3 millimeters. This species is found on the Oregon coast in the coastal rainforest. That's the front view if you were a little tiny insect and it was coming at you. These don't have, the harvestmen have no venom. They don't spin silk. There has been for years a thing on the internet where harvestmen or daddy longlegs are the most venomous arachnid, and on and on and on. But they don't have venom, and they don't have fangs for venom, so they're harmless. So another one, this species belongs to a group called Savicon, and if you notice these kind of fuzzy little pedipalps, the little leg-like structures front, they're like a little Velcro kind of hairs, and they can reach out and grab things and pull it into their mouth. This is a Caddo agilis. These are very tiny, about 1 millimeter to 3 millimeters body length, and fairly short legs for their size. And they're found mostly in the East Coast, New England, and they do come into the Midwest some. Very large eyes on these guys. This is a related one, the Agilis pepperella, or Caddo pepperelli. There's also some harvestmen called mite harvestmen, and they're very, very tiny, size of mites, and very short legs, look almost like ticks or mites, but they're actually belong to the harvestman group. And then this harvestman is an introduced species from Europe that is known from a few locations in New York, and most recently been discovered in Massachusetts by a friend of mine and myself. They're spreading somehow. This is another strange species, Dendrolasma, that's from the coast of Oregon. This is another coastal Oregon and Washington species. Notice the size of it. This is an eye of a needle. And the name of this is Paranonicus bruneus, and the name's bigger than the animal. This belongs to a group called Gonoleptidae. They're mostly the majority of the numbers of this group is in South America, and primarily great numbers in the Atlantic rainforest in the coastal area of Brazil. And that's actually more of that forest is gone and been cut down than what you hear about the Amazon forest being deforested and all. There's very little left of the Atlantic rainforest, but there's a great number of these really interesting opilionids live there. They're very colorful, a lot of patterns that are interesting, a lot of spines. This is one from Costa Rica. I didn't paint that on there. This one's from Trinidad, just an interesting pattern. Belonged to a group called Cosmetidae. It's got little spines with the little circles around the spine. Don't ask me why, I have no clue. If you like polka dots, this one's good. So this one's also from Peru. It's got a little spine on the back. This one's from Chiapas, Mexico. Has a very interesting enlargements on the hind legs. This one's from the Amazon in Peru. This is Costa Rica, and this animal is actually eating the head of an anta, a leafcutter ant, that you most likely just found it. It's probably the ant was dead, and they pick it. They do a lot of scavenging. They can attack and get small, soft-bodied animals, but not anything major and strong. This one's in Massachusetts. It's feeding on the end of an earthworm. Here's one that's got a katydid. I don't know if it attacked it and killed it, or if it just found it. This one in Chiapas, Mexico is eating some sort of vegetable material from a plant. This one's in southern Arizona, eating a tiger beetle. I accidentally kneeled on the tiger beetle, squished it, and all of a sudden, before I knew it, the harvestman was there eating. They can be cannibalistic. This one's feeding on another harvestman. This is up in Maine. These two are in Pennsylvania, and they're two very common, Leobunum vittatum. They're very common on the East Coast, spread around, and male is on the right, female's the larger ones on the left. And after the mating, the male will take its tarsus, wrap it around like a grapevine, hold on to one of the legs of the female, and the female wanders around looking for a good moist spot to lay eggs. And he'll guard the female from approach by another male. Here's a female with the ovipositor reaching down into the moss to lay the egg. This is one in Trinidad that's guarding, a female is guarding the eggs, and it has a few young that have already hatched. All right, mites and ticks. I'm running out of time here. This is a rake-legged mite I found in Arizona, but has some really nasty spines and all, where they will grab prey. Often you'll find mites on great numbers in beetles or in other insects. There's even aquatic mites that live in ponds. Here's a little red water mite. Anyone have an idea what this is? This mite, what this is? That's one scale of a northern water snake. So the mite crawled out from underneath that. And the famous deer tick. I'm gonna have to go through here quick. This is cyclosa. This is spiders right here hiding in the web. He puts all the dead prey items here and he hides in there, and so things can't pick him out. Dinopus, a net casting spider, builds a web, throws it down on an insect passing by. Theriosomatidae builds a little web, has a strand of silk going off to an anchor point and pulls on it, makes it a cone. As soon as anything touches it, he lets go and it springs back and catches and tangles up the prey. This is a female golden orb weaver. See the little spider there? That's the full-grown male. There's the male ready to mate. So while the female is busy with the food item, that's a leftover cricket there. While she's busy, the male will come down and mate. And I can tell you more about where this is. This was photographed in my shower. If you want to know, I can tell you a story later. Gastrocantha, spiny orb weavers. Meta ovalis, these are found strictly in caves or in little overhangs or hollows, sheltered areas. Orchard spider, venusta, Lacal venusta, excuse me. Look like a spider? That's the same thing. This is spider's unique in that it can actually bend and flex its abdomen, and then will just hang out, hiding, not visible to predators like this. Webs of all sorts. Nice spider on my finger, it's not biting me. Here's some ants that came out to start a new colony, so they start coming out, they crawl to, vegetation. They're crawling up here, bad location. Spider webs here, they're getting caught like crazy. The spider's having a field day. Some spiders, most spiders are individuals and will fight and kill each other and eat each other. But there are some social or subsocial spiders that live communally. And so this is several meters across, and probably hundreds or thousands of spiders living among here. Anolosomus is the spider. Here's what the spiders look like. They feed together, catch prey together. This is in Australia. You tip over, pull a piece of bark off, and you can see spiders hiding underneath there. This is a social spider in Australia that lives in small groups underneath bark. This is Philopinella. It's a uloborid that this was in Trinidad, and they build their webs in close proximity and live together. Tarantulas, little baby versicolor. This was in Peru. This is an African species. This was in Guyana, Arkansas. If you see a burrow that has a little bit of silk over the hole, you know that it's occupied. You know that the tarantula is down in there. They don't always do that, but if it has silk, you know it's down inside the burrow. This is a Malaysian trapdoor spider trapdoor. Here's the lid. They make silken strands that are trip lines that triggers any vibrations to them. They're sitting there. And if you flip that little lid open, there's the spider waiting. And it comes out in a blink of an eye and grabs its prey. Here's a different kind of trapdoor spider in Trinidad. Here's the lid. Here's just right at the entrance, looking down into the burrow. See how well made it is? It's got a beveled edge, watertight when it's closed. Even arachnophobes would probably like this spider. This feeds on other spiders. Method. This is in Guyana. Here's another species that attacks other spiders, sometimes steals prey from the other spider's webs. But here's one eating another spider. And this is a jumping spider. Jumping spiders are sort of the teddy bears of the spider world. This is a female Phiale formosa. That's the male. Males, just like birds and other animals, the male and female look totally different. You heard a little talk about the Mavia inclemens, the two different morphs, the gray morph and the black morph. Here's the black morph. Had the neat little punk hairdo, tufts of hair. This is also the same species. It's the other morph male. And this is the female. You see, if you saw the segment about where the eyes move, in the back of the eyes of jumping spiders. And as you look at them, the light, dark eye, that's the back part of the eye scanning and moving. So I think we're over. No?
We're a little bit past.
So anyway, we're near the end, so if you have any questions, I'll be glad to talk to you later if we can't do it now.
Great, thanks a lot.
All right.
We can put the Anybody like to ask about any of the photos? Okay.
All right, well, thank you very much.
Thanks a lot.
Thanks, Bill. I'd like a quick round of applause for all of our speakers tonight. Thank you so much. And thank you for joining us. We really appreciate it. This was a great set of talks. Thank you for joining us online. And that concludes the speaker series for Casual Night with Arachnids. Feel free to swing by out front. There's probably a few of the activities left going on. Otherwise, have a great evening. Thank you very much.